morning to everyone who has joined Breakfast at UMHealth. This is episode 5. Okay, so we're going to kick off this morning with the first presentation by Professor George Snounou. I hope I pronounced that correctly.
That's fine.
He's the research director of the Infectious Disease Models for Innovative Therapies from France. And his title today is Current Status of Malaria and Potential for Control. A very good morning, and I'm looking forward to your talk.
Good morning. Well, thank you very much for inviting me to present at your club. I'm very honored to be here and to give you an overview of what I think is the problem with, or an overview of malaria control today. Now, malaria is still a global challenge today, as it was 100 years ago. Although the situation is better now, we are still in the grips of a large amount of morbidity and mortality, which unfortunately hasn't been helped with the COVID situation. So So I would like to present to you an overall view of the parasite, its life cycle, and then follow on with talking about prevalence and epidemiology, and then control and elimination. Now, right, I think you all know that the malaria parasite is transmitted by Anopheline mosquitoes. The cycle is both deceptively complex and also quite simple. An infected mosquito bites a person, the parasites go to the liver. From the liver, some parasites escape after being multiplied in the liver, and then they go through a cycle in the red blood cells, and it is in that cycle that people have experienced all the disease. Part of that cycle is the production of, as you can see, gametocytes, which are the sexual stages, which another mosquito picks up, and the infection proceeds in the mosquito, the parasite develop, they go to the salivary gland, and the cycle is completed. Now, here are the landmarks of the allow me a second. Here are the landmarks that have characterized malaria parasite. It took about 100 years to discover the whole cycle as we know it today. Malariology has started in 1880, when the discovery of the malaria parasite was made by a French medical officer working in Algeria. I have put here the number of publications that published each year between then and now. As you can see, over the last 20 years, publications have increased substantially. Now, I can't seem to be able to move the slides forward anymore. I'm sorry, there's a problem with the slides. There. Transmission by the Anopheline took about 20 years to discover. The hepatic stage, an important stage in the malaria parasite because it's the first one, took another 50 years before it was discovered. And the hypnozoite, the dormant form that characterizes some of the parasites, was only discovered in 1980. One of the reasons why a lot of work has been done and publications have increased is because from the early 1980s, we could manipulate and clone and in vitro culture the parasite. And this has allowed us to discover a lot of what the parasite is doing and how it functions. And in recent years, this has been increased with a huge amount of data that's coming from the genomics and the transcriptomics, et cetera, et cetera. To summarize, there are 4 malaria parasite species that infect humans, and these 4 are apparently kind of similar to each other. Biologically, they are similar to each other, but they differ in the way they act, they differ in their pathologies, they differ in their transmission. And although these differences are subtle to the eye, especially when you look at the morphology, they are very important from the point of view of epidemiology. And one should always consider what species one is dealing with when you want to control the parasites. Out of the 200 or so Anopheline species that exist in the world, about 40 of them are considered as good vectors for malaria and important vectors for malaria. And as you can see, their distribution varies from country to country. And if you looked at that map, if that map was made 100 years ago, it'll be exactly the same distribution. The Anophelines have not changed in their distribution over the years. Now, the prevalence of the malaria parasite has indeed changed. If you have a look at this slide here, which summarizes the prevalence and the areas where malaria occurred, if you came here in the early 1900s, malaria existed throughout practically all of Northern Europe, the Mediterranean basin, the North and South Americas. It extended to Siberia, to Finland, to Sweden. Malaria is a global problem, and is not today, only today it has become a tropical disease. Now, understanding the malaria epidemiology is in a sense simple. It's a mosquito that bites a human who becomes a carrier, and another mosquito bites that carrier, bites another human, and then the cycle proceeds that way. It takes about 12 to 30 days for the parasite to grow in the mosquitoes, and one would think that this is a simple epidemiological equation. However, given the diversity of the anopheline species, of the parasite species, of the environment in which both anophelines and humans live, and the way the infection persists and lasts and produces the sexual stages for transmission, it is a very complex equation to solve. And indeed, one of the conclusions from a lot of the work that's been done on malaria is that if you want to understand an epidemiology of malaria in a particular place, it is local. You can go 20 kilometers away and the epidemiological picture would be quite different. This makes it difficult to control this disease, because when you set up control measures, you really have to consider the local conditions and for each of the places where you want to control it. Now, the other very important thing to understand is that practically all our knowledge from malaria and how malaria proceeds in humans comes from microscopic observations of people. And if I've put on this slide, the way one would see these parasites, at microscopy is limited to about 100 million parasites per person. If you go below 100 million parasites per person, then microscopy will fail to detect it. Therefore, if you look at a person who's been infected over time, normal observations will show us a few episodes that occur over the time. That's the perception we have. If we look at the reality of what happens, this is what actually is happening in these people. You have infections that are sometimes like this one recurring. You have new infections that appear and disappear. You even have some infections that are invisible, although they're to the microscopist. PCR has helped to a certain extent what we're seeing, but it's only limited to about 1 million to 10 million parasites per person. Anything below that is beyond our view. And in my opinion, it's what happens below the microscopy level which determines whether we understand how the parasites interact in order to understand how pathology, immunity, and transmission are regulated. And if we don't understand these, it's very difficult to then control the parasites efficiently. And indeed, if you look at today what we know, despite all the work that has been done, we know very little about the nature of the immune reactions that control the malaria parasite, because we do acquire immunity, but we don't know what this immunity, how it's acquired, and how it controls the infection. We don't really know why some people get ill and others don't. You have to remember that people are infected with malaria over a long period of time, but only for a very short time do they feel ill. And given that there are millions of people infected all the time, even a tiny proportion getting ill makes for high morbidity. And with respect to transmission, gametocyte production that pass on to the mosquitoes occurs at an apparently random way, and we don't know what regulates that transmission. Oops, sorry. So ultimately, in most endemic areas, one can consider that most people will have parasites in their blood, even though they're all asymptomatic. Many infections are undetected by PCR. PCR analysis has revealed that there are a lot of them, but it doesn't reveal the full extent of what it is. Indeed, the carrier population is extremely large, and I think people who are trying to control malaria today have finally taken this on board, and the concept of asymptomatic malaria as an important epidemiological factor has now become central to malaria control programs. Now, what are those control programs? Now, we are lucky in a sense that we've had a drug that acts against malaria for a long time, because in order to control malaria, you have drug prophylaxis and treatment, you have to reduce the man-mosquito contact, and only recently have vaccines started to become a possibility. So the drug quinine comes from the cinchona bark, as you know, which was discovered in Peru in the 1600s. And the first time this was brought into Europe was by a Vatican priest, Cardinal de Lugo, who brought it into Europe in 1643. At the time, quinine was extremely expensive and therefore wasn't used very much, but it was proven to be efficient by a variety of clinical studies done by Sydenham, in England, Tote in Italy. And in the 1820s, so it's about 200 years after it was introduced, two French chemists managed to purify quinine. And after the 1850s, in the 1860s, quinine trees were brought in, seeds from them were brought in into Indonesia. And between the 1880s and the time the invasion of Indonesia by the Japanese, quinine production was concentrated, was a monopoly of the Indonesian archipelago. So after the discovery of the parasite and its transmission, there were two lines of competing lines of strategies to control malaria. One of them was championed by Robert Koch, who said, if you treat everybody with quinine, then the parasites will disappear, the mosquito will have nothing to feed upon to transmit, and you can then eradicate malaria in that way. On the other hand, Ronald Ross, who discovered the transmission by the mosquito, said, well, if you eliminate the mosquitoes, then it doesn't matter, the infection will die out and nothing happens. And both of them tried their strategies in various places, and in all cases, it failed. And it's only a combination of the two that actually will work. And indeed, it's a combination of these two approaches, attack the parasite and attack the mosquitoes at the same time, reducing man-mosquito contact, which is the basis of our control strategies today. Now, in these studies done in the early 1900s, it appeared that it became clear that quinine was not really a highly effective drug. It is rather toxic. It is, even then, it was a little bit expensive and limited. And after the First World War, it was realized that somebody had to, quinine needed a replacement. And indeed, today, it is wars that have driven our search for malaria vaccines. After the First World War, the Germans in particular, and then followed on by the Americans and the Europeans, have been seeking malaria vaccines, malaria drugs every time there was a war. Plasmaquine and atropine came first, but these have been discontinued. Chloroquine was discovered in the 1930s and started to use after the Second World War. The same with the antifolates. Primaquine, which attacks the hypnozoites, came because of the Korean War, and the Vietnam War has given us mefloquine, which was useful for a long time, and the Chinese on their side discovered artemisinin, which are today the mainstay of malaria control programs. Hopefully we are still seeking new drugs today, but hopefully not because of a war, as because we need to control malaria. So, as I say, control of malaria was to do with treating people using larvicides, and I call it engineering because that's drainage, modifying housing. This was really how malaria was controlled until the 1950s. The two discoveries that have made it possible to consider eradications was chloroquine, which was a highly effective, very cheap, very few side effect drug that is used against all the malaria species, and the discovery by Müller of DDT, which was an excellent insecticide. Although these discoveries were made early, it took about 10 to 14 years before they actually came into use. And it's the combination of these two that fueled the first eradication campaign, which started in 1955 and was sponsored by the WHO. This campaign, unfortunately, eventually failed, mainly because of drug resistance, and it was abandoned. Today, we are in a similar situation. We rely on artemisinins, which were discovered in the early 70s by 2UU, and then by insecticide-treated bed nets, which were discovered, which were first championed by French scientists working in Africa, and then first demonstrated to be highly effective by Pedro Alonso working in the Gambia. And today's eradication campaign relies heavily on these two strategies. Resistance, as I said, is the major problem for malaria control. After the eradication campaign started, it very soon appeared that chloroquine resistance was selected, chloroquine-resistant parasites were selected. Similarly, DDT-resistant mosquitoes appeared very early on. And it's because of the appearance and the widespread of this resistance that the campaign failed to a large extent. Now, I have to say that when these resistances appeared, the WHO at the time and the scientists, instead of kind of modifying the hypothesis, tended to ignore them because they were a bit of a nuisance. All the drugs that have been introduced since the parasites have developed resistance within a relatively short time. This is now, unfortunately, appearing to be the same with the widespread use of artemisinin combination therapies. Parasites resistant to artemisinin are starting to appear and spread, and they've even reached Africa today. We're also finding that mosquitoes are becoming more and more resistant to pyrethroids, so the effectiveness of the bed nets is reducing. This is going to be the major problem to solve over the next decade or so, if we are to sustain malaria control programs as they are today. Vaccines are, in a sense, our hope, or has been the hope since the 1980s. There are, with the advent of molecular biological techniques, cloning, monoclonal antibodies, we have now discovered, we have now many targets that we can aim our vaccines on, be it against what happens in the mosquitoes, against the sporozoites and the hepatic stages, and these will be pre-erythrocytic vaccines, which will stop the infection, or against the growth of the malaria parasites in the blood, which will reduce both transmission and also reduce morbidity. Unfortunately, to a certain extent, we don't know very much about why malaria parasites, how immunity is acquired, but the mentality that fueled the vaccine search was that if you have that you acquire immunity with age, with exposure, and therefore that anything that is in the hyperimmune serum must be what is the basis of this immune reaction. And therefore, what people have done since the 1980s is to screen the proteins from the parasite. Any of these proteins that are recognized by hyperimmune serum were cloned, were sequenced. Today we just download them from the databases, and then you publish that these are potential candidates, and then efficacy trials are conducted. And to date, many of the lessons of the past about immunity have been forgotten, but that's a different story. To date, I present here a very small list of the number of malaria proteins that have been considered as antigen and tested in preclinical and in some cases clinical trials. There are dozens of them that have been tested and considered. And to date, after 40 years of search, only one of these proteins, the CSP, forms the basis of a vaccine that is just recommended by the WHO a couple of years ago in 2021. And this circumsporozoite protein, a protein that covers the sporozoite, which is the form injected by the mosquito, which has been packaged in particles with and a strong adjuvant, and is called RTSS for the repeat, the T epitope and the S antigen of hepatitis, which really is combined, which is now the vaccine that is used today, and a variant of it is called R21, produced by in Oxford universities. Unfortunately, despite all the studies that have been done and despite all the optimism, these vaccines do not prevent infection. They reduce the number of clinical episodes, and that's really a minor scratch on the morbidity of the malaria parasites, and not going to be highly effective for the control. People are still looking for vaccines, and we hope that it's unlikely that we'll find anyone in the next 10 to 15 years that will be effective and licensable. In Southeast Asia, as you all know, we have a major obstacle to malaria, to eliminating malaria, to call it eradicated. And these are the infections, the zoonotic infections that were found. Both of them were found here in Indonesia and described first here. Plasmodium knowlesi, which remains the main parasite that is in Malaysia today, and Plasmodium cynomolgi, another parasites of macaques that exist in Southeast Asia, and these macaques only occur in Southeast Asia. Therefore, in a sense, unless we can get rid of the malaria parasites in the macaques, Southeast Asia will always have a potential to have malaria endemicity. Now, I will leave you with one final slide. I'm sorry this talk was short, but this is very difficult to encompass all the things in such a short talk. But I will leave you with a view of what the future hope will offer. New drugs, we will hopefully find there are many of them being tried. New insecticides, similarly, although we will know that if we don't use them properly, resistance will develop. Many people are considering genetically modified parasites or genetically modified mosquitoes. In my views, this is kind of pie in the sky. Up to now, these genetically modified population work very well in the laboratories, but they are very difficult to implement in environment. There is, however, one known effective vaccines. If you remember the slide I showed you about the prevalence in the world in the 1900s and today, malaria has disappeared more or less by itself from the large areas of Europe, North America, Australia, parts of South America, the Mediterranean basin, and is now confined to the subtropical region. And one of the main reasons, and I think the main effective vaccines that you can have today, is tarmacking the roads. If you have access to the rural communities where malaria is mainly, the reservoir is mainly there, then you basically reduce the chances of malaria disseminating, because having access means that the economic health of the communities there, the economic health of the country will be improved. And indeed, if people's economic health, if a country's economic health is good and everybody can share in that economic health, malaria tends to disappear by itself, with the help, of course, of a public health system which targets the parasites and the mosquito. I'll leave you with this thought, and thank you for your attention. Thank you. I'm open for questions.
Thank you. Sorry, I'm Prof. Fong. I'm supposed to be the moderator here. I apologize for joining late because I had problem.
Not to worry.
Yeah. Thank you, Professor Snounou. Quite incredible feat, compressing 100 years of malaria into into just a half an hour. Those who are interested to get with Prof. Snounou, you can chat in the chat box. Yeah. Any questions from the audience? Put it in the chat box, and we can convey it to Prof. Snounou so that we can respond to your questions.
Apparently, I've been clear and not controversial. Therefore, there are very few questions.
Yes. So that's a question from Professor Lau. He said, what do you think is the best strategy to control zoonotic malaria, Prof.
So this is $1 million question. If we knew and we had a simple answer, then we would not have zoonotic malaria today. How shall I say? I mean, the best strategies would be to kind of keep the contact between the humans and the monkeys to a minimum, but that's very difficult, because that either it implies that you're going to deforest and get rid of the monkey population, or that you have to get rid of all the monkeys, and this is not viable. I think the best strategy to reduce it is prophylaxis. I think, because we know that the people who actually suffer and get infected with the Plasmodium knowlesi in particular, Plasmodium cynomolgi is probably a minor problem from the public health point of view, are people who work in the forests and who work on a regular basis in the forest. And in a sense, distributing to these communities Malaron, for instance, a kind of a very effective prophylactic, not very expensive nowadays, that it's not licensed anymore. I think that would be the best way to reduce the morbidity. I don't think there'll be any way to get rid of the problem completely and eradicate it, because it's impossible to treat all the monkeys and eradicate the parasite. So what I would suggest is to provide prophylaxis for those communities which are come in close contact with the monkeys. Might be the best way.
Thank you. There's another question that is posed here. I would like to ask, what is the reason, or how does the malaria
Well, people,
fine to the top of the region?
it's very simple. Well, it's very simple and complex at the same time. Malaria used to be highly endemic in Europe. In some places like Italy, malaria used to be nearly as endemic as it is in Africa, with a lot of mortality and morbidity. And this extended all the way to Holland, Greece, Spain, and Italy. The reasons they consider that helped with, number one, the public health system is good, and therefore people had access to early diagnosis and proper treatment. Second, many of the people were living in the conditions in which they were living, their housing, their proximity to what was improved to a large extent. As I said, tarmac road helped because that meant that access to health, to materials to build better houses was there. Plus, the drainage, they drained a lot of the marshes. They've engineered a lot of the waterways so that they would not have stagnant water, and that reduced the number of mosquitoes that are there. And because of better housing, of better drainage, the mosquito-man contact has decreased. And as the mosquito-man contact decreases, the R0, which is the reproductive index, we all know what R0 means today with COVID, the R0 decreased to below 1, and malaria disappeared by itself. And really, this is, equally importantly, after the reduction of malaria to very low levels, the strategies to keep it under controls were maintained for a long time. One of the failings of the WHO eradication campaign is when it started, after about 10 or 15 years, many governments, many places have abandoned it, have abandoned funding the malaria control programs, because if you had 100,000, 200,000 malaria cases per year, and it dropped to about 20 or 30, ministries of health were not going to spend a lot of money trying to control 20 or 30 cases. But indeed, it is these last 20 or 30 cases for the next 4 or 5 years, when they get to zero, this is when the money should be spent. It's easy to reduce from 90% by 90%, is the last 10% that are difficult. So that's what I think it is. It's economy. It's as President Clinton said once, "Is the economy stupid?" That's what it is. I have another question. See here.
I'm afraid I think we are restricted by time, because the next goal is. We really thank you, Prof. Snounou, for a really incredible talk, compressing everything in half an hour. There are still a lot of questions, but just to inform our member audience that Prof. Snounou is now in our Department of Parasitology as a visiting professor. He will be here until next week. He will present a talk at our department, and those who have questions can always feel free to come to our department to attend the talk. And also, I think it is also online, and there will be another opportunity for member audience or anybody who wants to meet up with
I'll be very happy to discuss and answer any questions. Anybody comes and
you can meet
at the Parasitology department
face to face. Yes. Thank you very much, Professor Snounou, for very enlightening talk.
Thank you very much. Thank you for your invitation and for listening to me. Bye-bye.